to introduce the panel tonight. And you can see their pictures, but I'm going to give them the opportunity to describe to you who they are and what they do. And we'll start over here first with Jonathan MacQuitty.
So a very long time ago, I have a PhD in bioinorganic chemistry, and I did some research, but I did an MBA at Stanford, and I joined Genentech in 1982. I worked at Genentech and Genencore, and then in 1988, I started my own company, which was focused on a number of things, including replacing the mouse antibody genes with a human equivalent so that when you immunize the mouse, you could make human sequence antibodies. Subsequently bought by Medarex and then by Bristol Myers Squibb, and the drugs that, oncology drugs and other drugs that BMS sells are made from that platform. So in '97, I moved into the venture capital business. I was in the venture capital area for 18 years. And then I retired very unsuccessfully, very, very unsuccessfully. I retired in January 1st, 2015, and within 10 days, literally 10 days, I'd met someone who dropped a dangerous idea in my mind. And I don't know how much time I'm supposed to spend on.
2 minutes max.
2 minutes max. So anyway, I'll tell you later, but that led to 47, and me agreeing to be a short-time temporary CEO. That was in, you'll notice that it was neither short-term nor part-time. So I'm now finally about to hand the baton off to somebody else. So he actually announced yesterday my successor, which I'm delighted about. So that's me.
Thank you. And then Karin Jooss.
My name is Karin Jooss. I started my career in the US in gene therapy, focusing on viral vector, immune responses to viral vectors at UPenn. From there, I then moved back to Europe, to Paris, where I started an immunology lab, focusing on studying immune responses to viral vectors. We try to make it long-term stable. And from Paris, I moved back then to the Bay Area. I was head of research at Cell Genesis, heading the cancer vaccine group, focusing very early on combination therapy, combining cancer vaccines with immune modulatory antibodies, which we published extensively during that time. From Cell Genesis, I then moved on to join Pfizer for a 9-year stint, where I was hired to set up from scratch cancer vaccine development. At the time, Pfizer actually out-licensed their immune modulatory components, and I built a program combining cancer vaccines with immune modulators and moved the first program last year into clinic, the year before, in prostate cancer with an extensive pipeline for 7 cancer indications. I left Pfizer a year ago to join Gritstone Oncology as the chief scientific officer. And at Gritstone, we build cancer vaccines targeting tumor-specific neoantigens. We identify the mutations within the tumor and build personalized cancer vaccines, which we aim for moving into clinical development next year.
Wow. Thank you, Karin. So, Ed Engleman?
I'm Ed Engleman. I'm a professor of pathology and medicine at Stanford Medical School, where I've been for almost 40 years, overseeing the Stanford Blood Center and also my own immunology research laboratory. I've always been entrepreneurial, and a number of years ago, I was a founder of a company, infamous or famous, called Dendreon, which developed the first and I think only cancer therapy vaccine called Provenge for advanced prostate cancer. I'm also a co-founder and managing partner at Vivo Capital, which is an investment firm that invests exclusively in healthcare companies. And most recently, I'm a founder of a brand new immuno-oncology company called Bolt Bio. And that's enough for me, I think.
Thank you, Ed. And Jackie Benson.
Hi, everyone. I'm Jackie Benson. I work for Johnson & Johnson. I did my postdoctoral research here at Stanford. I'm a PhD immunologist. In 2001, moved back to the East Coast and worked for Centocor, which is now really the biologics pharmaceutical development organization within J&J Pharma. So we actually discovered Stelara using the Medarex mice that you just heard about, and I was the lead scientist for that program for 10 years, and then led the discovery of gazelkumab, which is another monoclonal antibody that we expect to hopefully get approval for later this year. So I study autoimmune disease and biological therapeutics to manipulate and improve autoimmune indications. And then last August, I actually moved back to the Bay, thankfully, and I now reside within our Innovation Center, which is really just a mile and a half from here. And we opened Innovation Centers about 4 years ago as a way to interact with the ecosystem. So our large pharma companies can be quite complex, and so we tried to simplify that by really having a one-stop shop for folks that want to interact with J&J. We have a location a mile from here with essentially residents from all the major sectors within J&J.
Thank you very much. And I don't want to spend too much time on myself. You can look me up on the website of my company, Atreca. I'm an academic refugee. I'll say at least that much. I was a Berkeley professor who saw the light or the dark, depending upon your frame of reference, and left it all behind to start a biotech company, and I'm on my second company now. So over drinks later, we can talk about that decision because it requires some alcohol. But I never look back, never look back. So thank you, everyone, for joining me up here today. Because I was a college professor, I think the organizers felt that I would do a reasonable job at the next task at hand, which is in the next 10 minutes telling you everything you absolutely need to know about the immune system and immuno-oncology so you can appreciate the Q&A and the discussion. So without further ado, I'll start in. What is immunotherapy? I think we have to do a little bit of definition here in the terms. The first thing is, well, immunotherapy is a treatment that fights disease by, I like to use the word invoking the immune system. There are lots of treatments that rely on the immune system to mop up and do other things, but when you're actually tweaking the immune system to cause it to be part of the solution, that's immunotherapy in the broadest sense. Now, I think maybe for some of you, there's another step we need to take, which is, well, what's the immune system? What is it doing? And again, I apologize. I just want to get everybody on the same page here. The immune system at the highest level is really out to identify what is not you and destroy it. And when we say not you, that means things such as infectious disease organisms, pathogens, parasites, tumor, and dead parts of you, cells that get damaged, that sort of thing. So things that are not you. And this is important for our discussion about cancer immunotherapy, because that plays very much into how cancer immunotherapy even has a chance of doing what it does. Now, for those of you who are not steeped in biology, and I have to say, I'll speak for myself, but I'm thrilled to be here because I don't have a hostile audience that's going to hang on my every word and point out the inaccuracies. So I'm very happy about that. So I'm going to keep this at a very high level, but feel free to comment or stand up and throw your program at me if you don't like what I'm saying. The first thing to realize if you're not a biologist is, like every system created by natural selection, the immune system is very baroque, okay? There's no rational design, zero, which means the task of folks like us is to sort of shake the black box and see what falls out and do it again and try and connect the two things that fall out in some logical way, and eventually you build up a rationale. How baroque is it? It just has a few different moving parts, just a few. This is from a recent paper described. Each square in that diagram is a different cell type in the immune system. And this is a mouse immune system. Humans are basically the same. Careful here, we got some real experts. They're more or less the same. And if you're a lumper or splitter, of course, these are the splitters. But still, that's a lot of complexity. And it gets even worse in a sense because every single cell that interacts with another cell has, as you can see in this diagram, an entire range of proteins that it uses to connect and communicate and control the other cells with which it contacts. And this is just the cell surface. I'm not even talking about the soluble factors that are involved. I'm not even talking about the signal transduction networks. And honestly, there is something of a rationale at the highest level, but a priori, there is no firm engineering involved in this because it's all generated by natural selection. The key, however, is that the system has many, many, many layers of control. And it needs to do that because if it didn't have the layers of control, it might not do such a good job of finding what's not you, and it might actually start attacking you. And that's why there's so many layers of control. And that's also important for our understanding of how cancer immunotherapy works. So here's the simplified version of all that. Conceptually, there are two basic parts to the immune system. There's innate immunity, and those are the pretty cells up there that their job, they've evolved over time to recognize things that our ancestors routinely encountered. Okay, certain pathogens over and over again, certain parasites over and over again, certain tissue damage over and over again, and they recognize this. Okay, without any ado, they're very fast acting. Okay, the other side is the adaptive response, and this is the crowning achievement of vertebrates. Okay, because our immune systems can actually, over time, over a series of days, maybe weeks, learn what's not us at a finer detail level and target it more specifically. Both of these parts of the immune system are very, very important in fighting cancer. And so what does that look like when we do, another beautiful diagram here. And this will give me an opportunity to actually say a little bit about the different panelists, each of their areas of focus in their company or their research. So in the middle there is the tumor tissue in orange, and there are a bunch of different cell types. And it's very important to at least follow me this far in some of the details. That you can see the T cells, and T cells act as part of the adaptive immune system by learning to recognize things that aren't you. And they're taught that. Okay, but the first cancer immunotherapy drugs on the market, which is Yervoy, Opdivo, Keytruda, they all act by targeting this class of cells, T cells. Yervoy acts by getting rid of the regulatory T cell that's shutting down the T cell next to it, the blue one, the teal one, that's actually doing the killing. And then there are other drugs, like, as I said, like Opdivo and Keytruda, that actually cause the T cell to be a little more active than it normally should be, ignoring some of the signal that the tumor is putting out so that it can do some killing. So if I look at some other cells there, however, well, there are macrophages, which are some of those cells in the innate immune system which just eat things that are not you. And actually 47 is intimately involved in trying to target those cells. And then over there at the, the right side you see antigens. So these are things that are released from the tumor that the adaptive immune system is learning about so that it can target the tumor that has those antigens and destroy the tumor. And so Gritstone is involved in using antigens that come up in tumor to instruct the immune system, to instruct the T cells and the B cells what to kill. Now, they don't do that by themselves. That's one of the things I hope I get across. It's all this incredible crosstalk and network. You see something labeled a dendritic cell there. So dendritic cells are the cells that do the actual instruction with the antigens often. And actually, Ed, and in his research, and Bolt specifically, focus on targeting dendritic cells to allow them to be more effective at instructing the T cells in order to more effectively learn what's not you, in this case tumor, and kill. There are also other cells you see there, NK cells. And actually what my company does isn't even on this slide. This is this is from Bristol Myers Squibb, and they obviously don't consider B cells an important part of the immune response. But that's not true. Thank you, Jonathan, for that. So this is cancer, right? Okay, now I'm going to show you another very different slide. Okay, there's my very different slide. This is what happens in autoimmune disease. It's at a very high level the same thing. that in autoimmune disease, the immune system, both parts of it, the innate, the adaptive parts, both unfortunately start attacking normal tissue. Okay, they somehow, the control circuits break down and they start attacking normal tissue. Normal tissue is you. Tumor tissue is mostly you. The thing about tumor tissue, remember, we're these assemblages of trillions of cells that have to work together and through evolutionary selection have been forced to work together. But once something starts reproducing a little faster, it starts being, there's selective pressure to select for cells that can mutate to get around the controls. And so mutation creates slight differences in the tumor, and that's what we're trying to take advantage of. So if, in a sense, all these autoimmune diseases you know of that are really horrible, right? In fact, there are more people with autoimmune disease than there are with cancer. These diseases, rheumatoid arthritis, multiple sclerosis, psoriasis, some other horrible ones. A lot's been known about that. We still have them, so not enough is known. What cancer immunotherapy is doing, all of us in the field, no matter what we're doing, is we're trying to induce autoimmune disease against a tissue the patient doesn't want, which is tumor. So there's a huge interplay. And how much of an interplay? So one of the first drugs, as I mentioned, Yervoy, that targets a particular molecule in the immune system in order to cause it to be a little more active, take away a layer of safety control. A different version of that drug, Orencia, actually helps CTLA-4 do its job better and is actually used in autoimmune disease to suppress the attack. So autoimmune disease is really the goal here, except we want to control it and we want to, focus it just on the tumor. So that's the introduction. And so with that is sort of the background. I'll summarize here. So first you have to understand, I showed you all that cell network, okay? And all those proteins involved just for those two cells and just multiply that by the number of cells. Almost any cell type, almost any protein, any control point is a potential target for therapeutic intervention. You can just imagine the number of potential targets out there, okay? Combinations, therefore, of these approaches are probably going to be necessary to realize the full potential of cancer immunotherapy. We'll talk more about that. I'll ask some of my panelists their views on that. But my view is it's not going to be like a statin. Okay, came up with a statin, we're done. Hypercholesteremia is cured. It's not going to be like that. There are too many control points, too much tweaking that has to be done. Which also means that we're really at the beginning, I'll say 2 decades of work. Maybe it's longer than that. This is the early days of cancer immunotherapy. And after the panelists have done with Q&A, a friend of mine is volunteered to come here, Dr. John Sunwoo, who's actually a physician who treats cancer patients and can speak exactly to just how wonderful this beachhead is. And he'll get to do that in a little while. But at the same time, we have a long way to go, and there's a lot of work to be done. And there's a lot of work at companies, there are a lot of startups that are going to be involved. So it's a good time. It's a good time from that perspective. So the need is great. The markets, as you know, are vast. And with that, I think I'll start turning it over to my panelists, because I've spoken enough. So I'll direct a question first to Ed. So I said he didn't have an earpiece in, so I assume he was listening to what I said. I said we're beginning of a long journey in cancer immunotherapy. Do you agree, disagree? What's the science telling us about the trajectory here?
Well, if you compare the trajectory now to where it was 20 years ago, the trajectory is much faster. I teach a course on tumor immunology and immunotherapy at Stanford. I've been teaching it for almost 20 years, and I think one can judge the trajectory by how many people show up for the course. Until about 3 years ago, it was strictly graduate students who had to take it, and a few postdocs. And starting 2 years ago, there were not enough chairs in the room to accommodate the number of people who wanted to take the course, and that's because it really works. I think prior studies suggested that it could work or work very modestly. Newer studies suggest that it can actually be very powerful. But as you pointed out, we are essentially at the beginning of the rapid curve. In my lectures, I always finish my lectures by saying that as it looks now, virtually every patient whose cancer cannot be cured surgically is likely to be receiving immunotherapy as first-line therapy. And so that's an extraordinary change from where things were. So we can discuss all the possibilities and the combinations that you refer to that are likely to be in place over the next several years, but we're off to the races at this point.
More comments or?
Oh, absolutely. Please comment, Jonathan.
So one way to think about this is that in all of us, there is a battle going on between the immune system and various aberrant cells that could create cancers. And the immune system wins a lot of those battles. And we don't know they're winning the battle, but they do. But unfortunately, evolutionary pressure means that the clever cancers, will escape the immune system. And what the latest work that Ed is talking about is ways to deactivate the mechanisms that the cancers use to deactivate the immune system. So the cancer does something to try to, escape surveillance or to hold back the immune system. And what we've done is to take the brake off the immune system so it can re-engage with the cancer. And so this battle between the immune system and the cancer, this is going to go on for a long period of time. You have something here which has the capacity to mutate, to escape these things. And one way to think about it, it's a little crude, but I think useful, is if one thinks of another infectious agent, which mutates very rapidly, like HIV, this was not controlled by a single agent. It mutates too rapidly to do that. So if we think about the future of immuno-oncology, it's going to involve several different therapies used simultaneously to try to control using the immune system and trying to gang up on the cancer. And another advantage about the immune system that people here know, but I think it's worth mentioning, is that the immune system has a memory. And so it can remember what this cancer looked like. And one of the things we're seeing with some of these immuno-oncology is that if you treat a patient, they stay treated for a period of time. Now, we have a word for a patient you treat and they stay treated for a long period of time. We call that a cure. So this is good. So we're starting to see things where you have a long duration of response would be the technical term for it. Durability response. So we're entering a very interesting area, and we have a little bit of a foothold on it. The problem is the cancers, will fight back, are fighting back, and so we're going to need a number of different tools in the arsenal in which to fight cancer.
Maybe to add to that, also to your timeline, maybe I'm too optimistic, but I think over the past 10, 15 years working in immunotherapy, I think we have learned, with all the failures, for example, in the cancer vaccine field, we have learned a lot about mechanistically how the different pieces work together. So we know that CTLA-4 or PD-1 as a monotherapy works in patients who do have T-cell responses pre-existent against a tumor, which we unleash with the immune modulators. And now we are seeking therapies for the remaining 70% of the patients. And we know we need to trigger those T-cell responses, tumor-specific T-cell responses in these patients. In the meantime, we have learned exactly where CTLA-4 interjects in T-cell activation, where PD-1 plays. And therefore, I think now is the time to design combination therapies where we can actually rationally design these combinations to exploit the power of immune modulators with other therapies to hopefully benefit the majority of the patients and not just 30 or 40%. So I think, so maybe I'm too optimistic, but I think the timelines are probably accelerating right now because we have a much better understanding how to put these pieces together in combination to hopefully benefit the patients.
So I hear two comments there. One is, yes, combinations are going to be necessary to drive more efficacy because there's so many different points to tweak the system. But also, to your point, I like the HIV analogy quite a bit. Taking what could be an acutely lethal disease and turning it into a chronic disease,
Right.
Eventually something else will get you. I mean, that would be the goal of long-term advances in cancer immunotherapy, where we're constantly fighting the tumor fights back, we hit back with something else, and 20 years pass. That would be the goal, and I think that's something that is potentially attainable here. So Jackie, since you're at Big Pharma, and part of this, we're going to touch on the science, of course, but I was told that a lot of the interest here is the crossing of the science and the business aspects of what we do. So I'll try and make sure we cover some of that. And part of the question I have for you is, since Big Pharma often is driving commercialization of these drugs and therefore drives investment even at the earliest stages, I'd like to know J&J's perspective. Are we just walking through a bunch of knowns now? Is there more science to do? What are you looking for since you're actually out there looking for new things?
Yeah, thank you. That's a great question. So I think I can speak for J&J. We realized some number of years ago that we can't possibly do everything within our own walls, within our own scientists, and that there's an entire world out there of very brilliant academic and biotech researchers that we need to find ways to collaborate with. And it can't be in a very traditional sort of exclusive licensing kind of way. So we needed to adapt to the ecosystem and to find ways to partner. So, and that's what J&J Innovation is an attempt to do. So we have 4 innovation centers. One is here in Menlo Park, and the others are in London, Boston, and Shanghai, where we think that those are truly the ecosystems of innovation. And within each center, we have residents, as I call them, from the sectors. So I represent the immunology therapeutic area within pharma. I have a colleague who's oncology. I have a colleague who's consumer, so our over-the-counter products and device, et cetera. And we have folks there that can execute deals. And we try to collaborate in a variety of different ways. We have a venture fund that's actually one of the oldest corporate venture organizations. It's been around 40 years. And so those folks reside in the innovation centers as well. So we do everything from invest in small startups. We have a J-Labs facility, which we're basically the landlord, no strings attached. So there's a host of startups in those spaces, and I think we now have 6 or 7 of those around North America. So in some cases, we're just a landlord where we get to see what sort of the young upstarts are doing. And we hope that there's a match someday, but there's no necessarily commitment. We invest, and we also do your more typical sort of R&D collaborations. And we try to do that with anyone who's doing anything interesting to us. And that allows sort of the world to be our laboratory. That's sort of our mantra. And I think it's critically important because that's where all of the crosstalk happens. So, we tend to be very focused on the areas in which we need to protect, which is areas that we have products. But that might blind us to where the next innovation is coming from. And I think technology is the other component that I haven't heard mentioned yet that I think is worth mentioning. So we can sequence transcriptomes, we can do proteomics, and so making sense of all of that data is a true challenge. So how do you take all of those different layers of biology, collapse it together, and then logically make those rational choices of what are you going to combine with what? How do you pick which patients to treat which therapeutic? And those are critically important decisions. And so we collaborate not just with life science companies, but also with technology and informatics companies, because really that big data challenge is a huge one. I don't think that anybody truly has the answer to, but we're certainly not going to figure it out in our IT group. I have a PC, so we're not going to solve that one. And it's just really important for us to contribute to the ecosystem in a variety of ways. And I think it's fantastic to see the advances in immunotherapy. I don't think that our company actually has any of those drugs, but that's okay, because if the field advances, we all advance, and then we learn more about the biology of disease, which is ultimately, what we're here to accomplish.
Okay. So I actually want to follow up with Karin, because you were at a big pharma.
Yep.
And this gets to the need for startups and entrepreneurial activities, and we'll touch on, definitely get Jonathan and Ed's comments here as well. But why did you leave? Why can't Big Pharma do this? Don't they have piles of cash sitting out there somewhere? I mean, and all the scientists they need? Why did you leave a great job at Pfizer to go to a raw startup in the life sciences?
For me, yes, Pfizer was a good experience for me. I was there 8.5 years. I set up cancer vaccine development. It was tough. It was a lot of naysayers in a big organization. It took a lot of tenacity, which I have. I moved something into clinic that everyone was surprised was moved into clinical development. I think I left Pfizer at the time where my program was moved into clinical development. I saw it move, it was in good hands at that point. And when I had the opportunity to join Gritstone, for someone like me with 20 years of experience in cancer vaccine development, targeting neoepitopes, targeting something foreign in the cancer like a virus, was really an opportunity to really make cancer vaccines a success. We struggled in breaking immune tolerance. I spent 18 monkey studies worth of money at Pfizer to break immune tolerance, which we accomplished against self-antigens. And I just saw for a patient the future in identifying neoepitopes, mutations within each patient's tumor, and building a powerful vaccine, which we are able to do in a startup. And I do not think this cutting edge research would happen at a big pharma like Pfizer at a pace we need to move these into clinic. Gritstone was formed 1.5 years ago. We are targeting to file an IND mid of 2018, which is very aggressive. We move very fast. We every week make a lot of important decisions to move the program forward. This is not going to happen at Pfizer at that pace. It took me 6 years at Pfizer to move the program in prostate cancer into clinic, and it was not lack of hard working and strong teams behind it. It's just the big pharma way of doing things. Having said that, I think once we do move closer into clinic, I think big pharma does monitor the space. I was in Pfizer continuously monitoring the space in big pharma. We want to make sure to support cutting edge research, but I think the initial steps need to be taken in a startup environment to move it closer to clinic or into clinic and prove big pharma this crazy new idea is actually can work and can provide therapeutic benefit to patients, and this is when then big pharma comes in.
Okay, I will come back to you about individualized therapies, but I do want to get Jonathan to talk about why do we need startups?
Okay, before we do that, I'm going to defend big pharma because I feel, I know we have someone who can do that too, but I feel I can comment as here as well, and I would say the following. If you're a big pharma organization today, Wall Street's expectations in terms of your revenues and profits constrain how much money you can spend on R&D. And what happens here is that later stage projects, which are nearer to commercial launch, understandably, are going to hog the dollars. And so I think over the last 20 years, if you looked at the amount of cutting edge research that pharmaceutical companies can afford to do themselves, they have cut back on that very significantly. The more sophisticated ones have realized, the point Jackie made, which is, we don't have to invent all these drugs ourselves, okay? And so they've been very careful to nurture early stage collaborations, which don't cost, you don't have to run the lab yourself. You don't have to spend that money, but you need to maintain very close relationships with them. And I would say every single pharmaceutical company in the world today of any magnitude spends a lot of time on thinking about who's doing interesting research and how do I stay in touch with them and how do I connect with them. And the innovation centers, for example, at J&J are a particularly good example of that. So I think we have to be careful here to fault the pharmaceutical industry. The P&L pressure here, I think, has made it very difficult for them to do, cutting edge research. also the risk-reward ratio is not well geared for, for a large organization. If you're in a small boat, and if you don't row across the ocean, you will all die, okay? It can focus the mind wonderfully. I think if you're in a large organization and no one will remember whether you came into work to row or not, okay, it's a different kind of thing. And so the motivations there, both on the risk and reward side, I think are effective at driving things forward at a relentless pace, okay? So I think entrepreneurship is a very important thing. I would say that being in an early stage biotechnology startup is, it's not like being an IT startup. You have a small number of people, you're doing sweat equity. Unfortunately, when you start these things, it's like sort of flying a 747 from London to San Francisco. You kind of need to know how you're going to get across the Atlantic, okay? You can't rely on some, mid-air refueling halfway across to take you there. You have to have a plan as how you're going to do that, both a plan that's operational, but also a financial plan. How are you going to, to get the distance and the amount of money you need in order to get to clinical proof of concept, okay, to events which are significant. That it's a lot of money these days. It is not for the faint of heart, okay? When you start these things, the first round of financing was $100 million for Gritstone. Yeah. The first round of financing for 47 was $75 million. I mean, you have to have a concrete plan as to how you're going to go the distance. It is not a casual thing.
Very well said. Ed, your view as the quote-unquote investor on the panel, although Jonathan fits the bill too. Why startups? What do you look for? Maybe is a better thing.
Well,
in immunotherapy.
Remember, I'm a hybrid, so I actually oversee a research group at Stanford that is filled with graduate students and postdocs. And they're dreamy-eyed, naive, willing to take infinite risk and work for almost nothing. Extraordinary efficient way to make great discoveries. Think about that. Actually, for me, it's a privilege. It's a great privilege to be able to do this, which is why I've stayed there. And the quality of the discoveries that come out of a lab like that is really quite amazing. Now, the group that I work with, the investment firm I work with, Vivo, is not traditionally a startup funding firm. They shied away from that years ago and moved to a later stage. They're perfectly willing to invest larger sums of money. They have larger sums of money to invest. So they're not willing to do that. They do have there's a small fund called the Panda Fund that's affiliated with Vivo that is willing to take that kind of risk. But it's a risky business, and in the area of immunotherapy, which is now crowded, very, very crowded, the main advantage is that you have all of the large pharma companies, almost all of them, shopping for the next best idea with which they can combine their particular checkpoint program. And so that means that the exits for these early stage companies can come quickly, more quickly than in the past. So that's the carrot, if you will. But trying to start something from scratch today is very challenging, simply because in immunotherapy, at least for immuno-oncology startups, it's quite competitive and very, very crowded at this point. Still possible to do if you have something that's truly breakthrough, truly different, but it's harder than it was.
Like Bolt.
Yes, exactly. Just like Bolt. but it's harder than it was 2 years ago.
And so I think that's some of the takeaways here, right? It's compared to perhaps some of the more, information technology startups that you might be more familiar with. The amount of capital required is just a different order of magnitude. I mean, I'll put it out there. It's easily to get a clinical proof of concept, $50 to $100 million, depending upon whether you're lucky or not as you move forward. And that's just to get to the point where you might be worthy of an exit. And so obviously if you're
or the next round of financing.
well, if the exit doesn't come,
Right.
your investors want to
Yeah.
take another dip.
Right.
And so, you know, the investors, of course, to make a return on 100 million, well, you got to think about even larger exits, right? It's a very different game that people play in biotech. But we also heard that there's a lot of innovation going on right now in small companies, where those small companies can move very, very quickly, to Karin's comments, and which is why J&J supports so much of that ecosystem, because so many good ideas are coming in small companies. So it is a very virtuous circle right now, but to Ed's comment, it's not easy, and the initial rounds of funding are getting larger and larger and larger. It's almost, it's the rare company now that has a real chance at success without having raised at least 20-some million in a Series A, and more along the lines of 100 or 75. Atreca was 56. There are large sums of money being put to use immediately. I would substitute one metaphor for the 747, though.
Okay.
I sort of liken what we did at Atreca to riding the bicycle while assembling it. So maybe it's like bolting on the wings of the 747
It
as
is.
you're flying it. It's a little more like that.
It is.
So I want to, you know, we've talked a bit about the science and where that's going. We've talked about why this startup entrepreneurial ecosystem is important. Of course, at the end game, there are patients that are being prescribed these drugs and payers that need to pay for them, and they're very expensive. It is funny, one small anecdote, I had a Honda hatchback as my first car as a graduate student, and it was used, but new. It was, in today's dollars, it would have been about probably $25,000. And recently purchased for my daughter, who's in college, a Honda hatchback of exactly the same color. Looked as if it was the same basic design with, a little bit of steroids involved. And it was $25,000 today. Only it was 2 orders of magnitude more car. Unfortunately, in healthcare, you're getting 2 or 3 orders of magnitude better outcomes, but you're not seeing the price stay the same. And so I think that's something that, especially if combination therapies become more and more important, as something that's going to fall out. Something has to happen there, and we could spend hours talking about it, but I think it would be useful to hear, perhaps, I know because Jackie's embedded in an organization that has to think about pricing. What are the concepts around, layering on $300,000 annual treatments on top of one another in order to drive a cure in only 50% of people? That's a problem. So what's the thinking in J&J about this?
It's especially challenging with biologics. So, we're more, I think, used to thinking about combination therapies with chemotherapies, which are small molecule drugs, which, regardless of how they're priced, the cost to generate them is not nearly as much as it is for biologics, and certainly not for immunotherapy. So that's an order of magnitude different. So I think if we're going to consider a combination of biological therapies, cost is a significant concern. Safety is a significant concern. I think the autoimmune diseases have a very different safety efficacy tolerance than oncology does, and the time is different. So for autoimmune diseases, you're treating chronically, likely for the patient's life, which hopefully is many, many years, whereas oncology is really more acute. So pricing is very different in the world of autoimmune disease versus oncology, even though the drugs may not be very different because we're going after similar targets, but maybe with the opposite mechanism. But pricing is, obviously, as we know in the news, it's a big challenge. It's just one piece, I think, of the healthcare challenge. And it is something that we worry about, we think about, we wring our hands about for many, many years. And it ultimately comes down to value. So you have to demonstrate value to the patient, value to the payers, and value to the physicians. And that's ultimately what's going to drive what the cost ends up being. And how do you define that changes over time. So how we define it today is different than 10 years ago. It'll be different than 10 years from now. And it's going to change according to regulations as well. So it's anybody's guess what it's going to look like, even maybe 2 years from now, or next week, depending upon what happens. So it is a big piece of the challenge that we have, but it's only a piece. I mean, there's more to it. Access is another. You know, how do we ensure that all the patients have access that need it? Because they don't, and the cost is related to that, but that's not the only hurdle to access. So there's a lot more that we need to do.
Go ahead, Jonathan.
Yeah, I mean, this is going to be an increasingly difficult problem, both for within the clinical community, but also politically. You know, there are a couple of things that I think will filter in, which will mitigate the problem, but they sure don't solve it. The first is that I think we're going to see increasing use of stratification of patients, where you're going to select a subset of patients where this therapy is going to work. So you're not going to spend money on patients for whom this therapy is not going to work. So you're going to see, I think, people wanting to have stratification. You know, if the stratification costs $5,000 and the drug costs $300,000, it makes a lot of sense to stratify people, you know, if you've got an oncology patient, to stratify them. And I think we're going to see that. We're already seeing it, I think, in PD-1, PD-L1. So the other thing is that I worry, I come from the UK, as you might gather. So there's a committee there with the delightful acronym NICE. So it isn't very nice. What they do is that they calculate, based on what a human life is worth, whether your drug is priced appropriately or not. So, you know, it is incredibly detailed analytical work to do that calculation. But of course, to some extent, the logic is hard to avoid. I mean, you know, if you're a payer and you're going to pay for things, you want to justify doing it. And this kind of analysis is the way that I think people are going to look at it. Does it make sense to have that price for this kind of problem?
So you made a very nice point there that I want to follow up on, which is, you know, we've been talking a lot about therapeutic approaches.
Yes.
But there are a whole slew of other things that are going to be necessary to realize the full potential, and part of that realizing the full potential is driving the cost down by treating the right patients. I wanted to ask Karin, because you're one of the people who's actually put something into the clinic. So what are some of the things that you're thinking about now in terms of biomarkers or patient stratification or other things that you know, are not just the therapeutic? Because that also provides an opportunity for entrepreneurship as well.
Yes, very good point. I mean, since we at Gritstone look at the tumor carefully, at the patient-specific tumor, to identify the neoepitopes that we need for our vaccine development, beyond the neoepitopes, we dissect the tumor signature. And in early clinical development, we will learn which patient will respond to our therapy and which one will not. Yeah. And so I think our database on what's happening in the tumor before we treat and after we treat, we take biopsies, I think will lead to patient-specific therapies that will then hopefully lead to benefit to these cancer patients beyond the 1 or 2 months that we have been seeing in the past, and where we go more for long-term durable benefit. So if you think about, since that Pfizer moved a prostate cancer vaccine into clinic, if you look at the healthcare cost to treating patients with castration-resistant prostate cancer, is huge. So if you prevent that from occurring, I think the price benefit is probably in favor of maybe the increased cost of a biologic. So we need to get beyond the 1 or 2 months survival benefit at an earlier stage, and then kind of eliminate the downstream cost to the healthcare system.
So before we sort of end our Q&A and to bring John up here and open it up to you folks, just wanted to canvass the panel here. Are there any comments, you know, that weren't elicited by my questions that you'd like to bring forward to the audience?
I would just add on the pricing issue that it's inescapable. The really expensive therapies, and when I say really expensive, I mean the cost of goods, not the price.
Yeah.
The price is determined by what the seller hopes they can sell it for. Cost of, if you have the same size fits all, if you have an antibody or even a small molecule or a biologic, take an antibody, checkpoint antibody that's going to be used by millions of people, the cost is really relatively small, and that price is going to be determined by competition, and it will come down dramatically. The real problem becomes with individualized therapies. I say to my partner on the right, especially cell-based therapies. I lived through that with Dendreon, and now we're about probably to have a CAR T therapy approved. The first company that to likely get approval has indicated they expect to charge, I think they said, between $600,000 and $700,000 per patient.
So just so everyone's aware, this is a therapy that takes a patient's own cells and engineers them and puts them back into that patient. So the therapeutic is only useful in one patient at a time. And each therapeutic needs to be made for each patient. And some of what Gritstone is doing is also similar in looking at patient-specific neoantigens, if I get it right. So just wanted to give that context for why would something be so expensive? Well, someone's making a drug just for you.
Well, the point I was going to make is that I think that these types of therapies that are truly individualized are a transient phase. That then science is moving very rapidly, and that over the next 10 years, these types of therapies may prove to be very important in the marketplace, but it's not going to last. And that we will figure out ways to target and manipulate the cells in vivo, in the body, using other approaches that are far less expensive, and that is the future. I point to one example, which may not be the only one, but when CARs were first described, one of the first success stories was in chronic lymphocytic leukemia, which is a relatively common indolent disease that ultimately becomes much more serious.
Yeah.
And there was a wonderful paper published in 2011 describing how an individualized CAR could kill those cells dramatically, and everyone was abuzz about this is the future treatment for CLL. And then a year later, a small molecule pill that could be taken once a day that inhibits a particular enzyme in the leukemic cells was developed and took 90+% of the market, as well it should. So I think that what's going to happen is that competition is going to play an incredibly powerful role here, and discovery, and we will eventually get past what I think will be a temporary phase, albeit a critically important phase.
Well, thank you very much to panelists right now, because I'd like to talk to have someone speak to you who's in the trenches. I think that's very important. Not that we're not in our own little trenches, but someone who's seeing patients, who's seeing the difference that patients can make. So John Sunwolf, please come to the lectern. John is associate professor. John is associate professor. I can almost never get this word right. How do you say it, please?
Otolaryngology.
Thank you. And had a neck surgery at Stanford, and is involved in his research in immuno-oncology, as well as being intimately involved in the treatment of patients with these agents. So John, I don't know if you want to.
Yeah, well, thank you, Tito and Brian, for inviting me, and it's been a very interesting panel discussion. And Ed has been a mentor of mine since my first month at Stanford, so it's an honor to be here. So as Tito mentioned, I'm a head and neck surgeon at Stanford. I deal with malignancies of the head and neck, and I focus primarily on melanoma and thyroid disease. And my lab is focused on cancer immunology, and specifically with an emphasis on natural killer cells, one of the other cell types that Tito mentioned. And in our field, September 2014 was a very important month, because that was really the turning point when the enthusiasm for immuno-oncology really took off, and that's because that's when Keytruda, or pembrolizumab, was approved by the FDA after just a phase 1 trial, primarily in advanced stage melanoma, and they saw remarkable responses in that trial. Not everybody responded. About 20% of people, or 20 or 30% of the advanced melanomas responded. And so that actually begs another question that was alluded to by the panel, in how can you predict who's going to respond, and how can you monitor that response? So there are a lot of, I think, questions that are really open, not just in predicting the response, but once somebody goes on therapy, how do you monitor? Is there really a response going on, or do you have to wait 6 months before switching them to something else? I think that's a really important question that's not really well worked out and is ripe for innovation. Earlier today, I was doing an operation. I wasn't sure I was going to make it on time. It was a gentleman who had a very large scalp melanoma and a large neck mass that needed to be resected. And by definition, he would be stage 3C melanoma. And these patients have about a 50 to 60% chance that over the next 5 years they're going to recur with disease, not necessarily in the primary site or the site that was operated on, but somewhere distantly. And so when that happened just a few years ago, and I remember this very well because I had a lot of patients who had this happen to them, they would not have a whole lot of options. There wasn't really anything very good out there for these patients. And now with pembrolizumab and nivolumab showing remarkable responses, I have a lot of patients who recurred distantly but then had a great response on these anti-PD-1 antibody checkpoint inhibitors and are alive today many years after recurrence. So it's been a real game changer. The next phase is really in which situations do you give it after surgery? When they have no measurable disease, they're considered to be disease-free, can you give them an immunomodulatory agent and prevent that or reduce that percentage from 50% down to 5% or 0? Is that possible? And I think there's currently a trial going on right now comparing dealer's choice interferon or ipilimumab versus an anti-PD-1 antibody. But then I think the other phase that's really interesting to me is there are some patients who respond dramatically after one dose of an anti-PD-1 antibody therapy, and their tumor responds dramatically, clinically, radiographically, and can you predict that? And so what would happen, we're doing a lot of research right now where we give the antibody preoperatively a few weeks before surgery, really just for research, so we can understand the immune cells inside the body, but the patient's going to go on to have surgery. But can you now maybe transition therapy before surgery? And this would be a total paradigm shift. You give it before surgery. If they have a remarkable response, maybe you hold off on surgery and you continue to follow them, and you see whether or not they actually need to have surgery. Maybe they'll have a complete response. And anecdotally, that does happen. We will give a dose of a drug and do the operation, take out what we thought was a mass, and there won't be any viable tumor cells there. So there are a lot of areas, I think, for clinical research. I think the challenge is going to be, with all these new agents coming on board, how are you going to prioritize which ones get studied? Because a phase 3 trial takes a lot of money and a lot of time, and with a whole bunch of tools in your toolkit, how do you prioritize? Which ones do you combine? Which ones do you try first? And then if you keep combining things, eventually you're going to get to that thing that Tito mentioned, and it's autoimmunity, which we do see. And it is pretty devastating, sometimes worse than the cancer itself. So those are just some comments I wanted to make. Thanks.
Thanks very much, John. So now we turn to getting some questions from you folks and other folks who are streaming this on the web. And I'm very happy that I don't actually have to answer these questions that I have in front of me. That's going to be their job instead of mine. But what I will do is I'll perhaps pick out some certain orders here, and there's some great questions here. Some of which we've already touched on. I think so. Something that I'm sure would be very interesting to this audience that I think Jackie touched on, and perhaps then somebody else here on the panel can pick up on. What are the top big data analysis challenges in immunotherapy translational research? What are the, and this is, I think big data and machine learning has yet to make the slightest impact in immunotherapy. So it'll be interesting to see what these folks say.
I'll start because this is
Okay.
something that I'm quite passionate about. So the past few years, I've actually been leading an internal venture within J&J focused on lupus. So I don't know if you know what lupus is, but it's an autoimmune disease. No two patients are alike. And it's just been riddled with failures for drug development until recently there was a drug approved, first drug approved in 50 years. But so huge unmet need. We think we understand a lot about the immune system. At least in our company, we have this portfolio of immune modulators. How are you going to approach lupus? And so we invested in actually non-clinical studies, just collecting samples from lupus patients over time, sequencing their DNA, sequencing their RNA, doing autoantibody specificity profiling, and then we got all this data. So then we needed to put it together and try to visualize it, understand it, and make decisions on it. And that's still a work in progress. But I think it's critically important. I mean, that's one autoimmune disease where we're going to have to identify the subset. We can't just take everybody that has lupus and put them in a clinical trial. We need to know exactly which mechanism our drug modulates and then match that to the exact patient. And I think it's very similar in oncology where you need to match the tumor with the therapeutic approach. And so the big data challenge, it's universal. I think I'm giving you an autoimmune example. We can talk about oncology examples as well. But that's, I think, where some real innovation can happen is how do you manage the data? How do you take layers of biology and make sense of it? And we have a couple of different collaborations. Some of them are in the Bay Area here with just topology tools. So how do you visualize data so that a dumb biologist can understand what it means and then make decisions about what patient subsets to track? I think that's a huge need, and I think it could truly revolutionize medicine across many disciplines.
Anybody else want to tackle the big data question?
Well, over the last several years, I had a group of students in the lab who were asking the question, simple question, how does cancer immunotherapy work? We don't truly understand. And they narrowed it down to a question, well, is it only necessary to take the tumor and change the environment in the tumor to make it more immunogenic, to change the makeup, cellular makeup inside the tumor, or do we have to change things throughout the body? And they did a very complex experiment that was published 2 months ago in a journal called Cell that addressed this question for the first time. And it was a complex data set, required development of new algorithms to analyze all the data, because what they did was to analyze the immune response, all those immune cells, 92 different immune cells in 6 different tissues, multiple different tissues, in animals. You can't do this in humans yet, that received either a curative therapy or a therapy that didn't work, and then compare what happened system-wide and inside the tumor. And the results showed that while you could change the tumor with an ineffective therapy, it didn't cure the animal unless you treated the entire immune system. That was rather surprising, but it reminds us that cancer is a systemic disease. And if we're going to learn how to use these therapies more effectively, we have to understand the response throughout the body, not just in a single targeted tumor and other stuff. So I think that we're really just at the beginning of understanding how these things work.
So here's another big data question. So if you take a tumor and compare it to normal cell, there are literally thousands of different mutations in that cancer compared to normal cell. Now, most of those mutations are synonymous mutations. In other words, they don't really change what the tumor expresses in terms of particular proteins. But there are hundreds of those, what are called non-synonymous mutations, where they're making a change which actually results in a compound being made by the tumor which is different, okay? Now, what happens here is, for various reasons, we don't understand why some of those elicit an immune response from the immune system and others don't. Okay, so what you want is a system that will work out which of those, maybe 25 or 30, which of those will be really important to elicit an immune response. And then you make a vaccine of those, okay? And the patient goes, hey, that's a cancer-making stuff. I don't like that anymore, and I'm gonna go after and attack it. But to do that, you have to, you've got to collect enormous amount of mutational information. Then you've got to work out which of those lead to changes being actually produced. And you've got to work out, quite trickily, which of those are actually going to elicit a response. All of that takes an enormous amount of big data.
Actually, maybe to add to that,
Yes.
since this is what Gritstone is all about, it's database and building an algorithm. Currently, what's being done in neoepitope-specific vaccine companies is that we identify the mutations in the tumor, and then what everyone is doing is using a program called NetMHC, which we use in immunology for how many years, where a peptide is being matched to the HLA about its probability of binding, and therefore we assume that this is being presented by the tumor. And we learned by doing some early analysis that if you really look at the tumor's HLA molecules on the surface and pull off the neoepitopes that are actually being presented, it's less than 5% of what's being predicted. So if you make a neoepitope-specific vaccine of 20 neoepitopes, 50% of patients would get no benefit of your vaccine. And so what Gritstone has embarked on is we have a large clinical surgical protocol open where we collect hundreds of tumor tissues and we actually do mass spec analysis, pulling the neoepitopes off or actually any epitopes presented by the patient's HLA. We get about 10 to 12,000 peptides pulled off of any given tumor, and we feed the information of the sequence of these peptides together with the patient's HLA makeup into a machine to formulate an algorithm for us that allows us in clinic, when we get a small slice of impure tumor tissue where we cannot do mass spec analysis, that allows us to predict actually the neoepitopes which we will build a vaccine for. So I think we are now into 150 tumor tissues being processed. By mid of this year, we probably have one of the largest databases in the industry to help us identify which neoepitopes are most likely being good T-cell targets, because we need them presented on the HLA. And I think, the machine learning is now available to us that help us actually to build these algorithms. Yeah. So I think the field has moved forward to help us assisting in building these algorithms.
Exactly.
That's great. So I think there are several different, if I can do a takeaway, I think there's several different levels at which a big data approach, whatever that means, couldn't be impactful. Definitely, I think part of the problem is, as you outlined it, Karin, is we haven't had big data sets yet. And I think it's only now that with a lot of the automated sequencing and effectively automated mass spec that you're doing, that we are building the terabytes and terabytes of data that will be useful for training neural nets and things of that nature. And actually, I mean, part of it is that biologists, you understand, tend to be hypothesis driven. We haven't yet embraced perhaps the fact that there are features in the data set that we don't know are important because we don't know how to ask the question yet. And that's, of course, what big data can teach us. So I think that that's something that you folks out there could, at some point, once the data sets are available, could help us with. A couple other questions here. I guess we touched on the role of biomarkers. I think that's fine. I think we also touched a little bit on a question someone's asking, is there going to be an aha moment for immunotherapy in the way that CRISPR was for gene editing? I think you heard from John
we already had it.
We've had it, okay? And I mean, you have to understand, though, there's a little bit of a backdrop. I mean, Karin commented on this. I think Ed commented on this. There have been decades of scientists in the wilderness believing that the immune system could somehow be useful in the treatment of cancer. And like a group of undesirables at a party, they were sort of shuffled off into a corner, into their own society, and they weren't really taken seriously for a very long time. And as you all are familiar with Thomas Kuhn's vision of, the old guard has to die before the new ideas can come. Unfortunately, the Young Turks, when this finally worked, are in their 60s and 70s. It took a while. But it was the breakthrough of pushing, and it was a real good combination of folks who never gave up the fight, who eventually got an immunomodulatory agent starting at a university, more than one probably, but we'll pick on one thing I have in mind now. Moving it to a small biotech company because that's where someone could take a chance, and then that being purchased by a large pharma company who took it the distance and showed that it actually would work. And that took a long time. But now that it's there, as Ed says, the curve is very, very, very rapid.
Yeah, one thing that, I don't know if people picked up on it, but if you think about melanoma treatment, 15 years ago, you had a bunch of cytotoxic agents. These are, they kill cells. They kill pretty much all cells. You hope they kill bad cells more than they kill ordinary cells, but they are really not very selective kind of agents, and their effect doesn't last for very long, okay? The cancer learns ways to fight around it. The thing that's really, I think, was very surprising in September 2014 was not just that 20 to 30% of these melanoma patients did well, but they kept doing well. You have patients who are now surviving for relatively long periods of time. You had durability of response. And generally we'd never seen that in melanoma. Is that fair to say? Yeah. And so that was at this point in time, people woke up and said, whoa, this immuno-oncology, this immunotherapy approach is really profound, okay? That was, I think, an aha moment.
Yeah, and you all know something, you all have a story right in front of you. Jimmy Carter, right, was in the news, right? And he had metastatic melanoma, Mets in his brain. 5 years ago, he would have been told to put his affairs in order, right?
Yep.
And he was 90, right? So how aggressive can you be with somebody who's that old as well? And he was Opdivo, right? And he got Opdivo, and he's cancer-free. He's one of the lucky 30% or whatever, but these things actually do work when they work. So we've had our aha moment, and that's why the money has been pouring in. And maybe that gets to one of some of the other questions here in terms of, it's a little testy, this question, which is, sure, there's a lot of innovation in the science and the technology, but I don't see a lot of business model innovation. I don't know is that something that the panels want to
maybe not in terms of the structure of the model, but keeping an early stage company going on other people's money for extended periods of time, okay, it's not like your positive cash flow. It takes forever, okay? If you sit down and think about it, even for an oncology drug, you're talking about hundreds of millions of dollars to do the work and get it through the clinic. It's just really, it's hard. And so you are constantly trying to persuade people that there's a pot of gold at the end of the rainbow, okay? You are, fortunately the pharmaceutical industry, I think, has been remarkably flexible about collaborating with early stage companies. I think that they have been, they understand to some extent the biotech and the farm team. They need to collaborate and work with them. That, by the way, is not always true. There are other parts of the healthcare industry where the large companies are not very supportive of innovation in smaller companies, but the pharmaceutical companies have been very supportive of that.
Anybody else want to comment on?
Maybe the support of big pharma also comes from the early days where PD-1 and CTLA-4 did show single agent activity. There was a lot of hype, a lot of money flowing into the space. And so if you look at the big pharma players who do invest in immuno-oncology, they have very similar portfolios in the immuno-oncology space. And now combining the immune modulators hasn't really panned out to the expectations of a couple of years ago. And so they are definitely looking for a differentiation in the immuno-oncology space. That means everyone is looking for something, what can I use to combine with my PD-1 or CTLA-4?
Yes.
And that probably has triggered a new wave on of investments into flowing into startup companies from big pharma to hopefully be on top of the race and combining it with the right thing to provide therapeutic benefit to patients. I think there was a second wave coming after the PD-1, CTLA-4, then the LAG-3, PD-1. So the new combinations didn't really pan out as well as it was expected or hoped for.
Yeah, I think it's as Jonathan commented, the business model, if anything, it's become more capital intensive, and but the basic bottom line hasn't changed. It's as efficiently as possible having something innovative show positive effects in a very diverse group of lab subjects called human beings. And it's just a very tricky proposition. But to Karin's comment, really, and I think Ed said this way at the beginning, real innovation still is rewarded. That you're not, if you're thinking about starting a company now, for an approach that's already in the clinic, I would say forget about it. That's my personal perspective, that unless you have something, and I showed you, there are a lot of different pinch points here. There are a lot of different places for somebody who knows the science to come in and make a difference. You're looking at folks here, at least the first four of us here, each company or lab approach is different, one of those different pinch points. So more capital being deployed, but it's in some ways the same old in terms of moving it forward to the clinic. But to get those companies off the ground requires innovation. And I think, I don't know, I put my view, less me too and more searching for innovation is what I'm seeing now compared to other like specialty pharma plays and other things in the past where too much capital was chasing the same idea.
I think there's, there are areas where I think that there are a lot of competitive activity. I don't know how many CAR T companies there are, but there are quite a few different CAR T companies. So there are areas which are crowded, but at the same point in time, we got a little diagram of all of the different things. So no, there are a lot of different clever approaches, and we're going to see people identify clever approaches, in the years to come. It's a crowded space, but Bolt is, there's room for a smart, as there is in other areas. There's a room for a smart idea, no question.
If I could just make one comment, I think the pharmaceutical development process is regulated, right? There's certain steps that we have to go through, but where I see a big sort of catalyst coming is the patient or consumer influence of into their own care. So there's much more interaction. Patients are much more informed, and they want to interact more about their care rather than just take someone's word for it. So I'm not sure what it's going to look like. There's a lot of different, health technologies out there, but I think the intersection of the patient being in control of their information and the sort of treatment therapy that they're on, and that intersection, I think, is where there's going to be truly new business models and revolutionize how we treat patients. I don't know what it's going to look like yet, but that's sort of where I see things heading, specifically in the Bay Area, because there's a lot of intersection between the health tech companies and the life sciences. So that's where I would look for some interesting catalysts of change.
Somebody did touch on this, and it's related to a question here. Lots of innovation on what to try and target, but at the end of the day, you have to do the clinical development, which can be a bit of a problem. If you were thinking about doing clinical development in melanoma right now, my understanding is forget it. You just can't get the patients too easily.
I don't know. There are 70% of those patients who still don't do so well. So, we don't know which ones they are. So, I think there's still plenty of opportunity, to treat patients who don't have good therapies. I don't think we're.
Right. So I'm sorry, I wasn't directing it too much to that as I was operationally getting patient enrollment, is a challenge in immunotherapy given there are 1,100 different drugs in development. So I guess the question here was more along.
Very crowded.
Yeah, what's being done? Can anything be done about some of those choke points in development? Not that there aren't patients that could definitely need the help, but getting the proof is a bit of a problem.
Mm-hmm.
I don't know, Ed,
well, it's a big, the good fortune and the progress that's been made does create a problem. The largest companies, Merck, Bristol Myers, Roche, and so forth, they are individually paying for and performing hundreds of clinical trials with their particular immunotherapies in combination with other components.
Hundreds.
I think Merck alone is up to 400 clinical trials. Now, think about that.
Yeah.
Each one of those trials is
Roche,
many
Roche
millions of dollars.
Yeah.
So it's true. And also now that patients are receiving checkpoint antibodies routinely, if you want to try something new, you have to try it on the background of patients receiving that standard of care. So it is, in fact, somewhat more complicated, but doable.
Karin, you had a comment?
I think it's doable in a way where you, in the very past chemotherapy trials, you opened a lot of centers and went into your clinical studies early on. I think for immunotherapy, especially if you go in combination with multiple modalities, you handpick a couple of centers for which you have very good relationship with the investigators, and then let them try at least the first in-patient study. Also, what's being complicated in immunotherapy, that for us to learn in a first in-patient study, we often want tumor biopsies throughout the therapy, as well as blood PBMCs. We often ask the centers to process the blood for us, which is challenging, to do immune assays to understand whether the vaccine works or PD-1 works. So the complexity of these early clinical studies usually limit us to really select a handful of centers to really show proof of concept. It's not going into clinic with 30 clinical sites any longer.
That's a really good point, and one worth making more generally. Again, 15, 20 years ago, clinical trials in oncology were run in a fairly standard phase. You were looking for clinical endpoints, how the patient, disease of the patient really got better. Today, we're focused really on translational endpoints. So those are endpoints when we're not looking so much as to whether the patient gets better, although that is certainly a thing, but we're doing a lot of very sophisticated imaging of the tumor. We're looking at, what are the immune cells within the tumor? Did they penetrate the tumor? Which subset of immune cells went into the tumor? What was the impact on other parts of the body? So the amount of analysis that's done is just extraordinary. So each of these patients, the biopsies and the blood draws and things like that, the level of research analysis is much, much higher than it used to be.
So I think we have time for one more question. Thank you very much, everyone. And if obviously there are more questions here than we could have handled. And those questions that are directed to somebody in particular, such as, why the hell aren't you doing this? You should just corner them after we're done here and throw them against the wall and ask those questions. I'm sure they'd be happy to answer them. I'm joking. It's not that bad. But the comment, this naturally feeds in, I think, to the last question. Now I'm going to go to Jackie for a first answer here. We've heard about the FDA and how, used to be, it seemed the FDA was more interested in covering their behinds in case they were dragged in front of a congressional committee if someone died. Seems as if there's been a change in how the FDA is viewing especially oncology drugs. And so I guess there was a question here about, well, is the FDA helping or hurting, or what's going on in the regulatory environment? Because that's sort of the last step before getting these out to patients.
Yeah, I think it is changing, and I think it's getting more facile, but it is a regulated environment, so there's only so much they can change because patient safety is paramount, and that can never change. But they are recognizing different designations. So there's fast track designation, there's breakthrough designations, and so that allows them to manage their resources so that they can truly fast track a drug that looks like it truly has a magnificent impact where there's a very high unmet medical need. So not a me too, but something that's truly transformational. So at least they have defined sort of categories and criteria that allows those sort of expedited reviews, but you still need to do the studies, you still need to have the data. And that shouldn't change. I mean, we really do need to be mindful that these are very brave patients that participate in clinical trials. It is not easy. It is a sacrifice on their time and their family's time to get them to where they need to go for all of the site visits, et cetera. So we need to respect that. So I think the FDA is trying to do what they can within the parameters that they must and we must operate within. But I think it's better. I think it's recognized that in certain diseases, we do need to be faster on how quickly we evaluate the data and have the sort of statistical models built in so that we can see that there's data to be looked at. So, you know, all the different analyses that are run, we need to, and we are improving in what the statistics are that really are those triggers, that this is something magnificent, and that we really need to take a look sooner. And we're doing that within our own clinical trial teams, but the FDA is doing that as well. So I think we're getting there. We're not where any of us want to be, but there are very important parameters that we need to work within.
So one last comment from anyone on this topic here?
Well, I think on oncology, I think the FDA is doing actually a very, very good job. You know, they are allowing people, as we heard, to get approval on relatively small studies. They're understanding that these patients are really sick, and you know, they're being very flexible about how to deal with that. And I think there is a contrast between, you know, oncology, where you have patients who can often be very sick at risk of mortality, from how they treat, you know, chronic conditions, autoimmune conditions, where obviously they're going to be alive for a long period of time, and the safety issues become different.
Well, that's it. We're, unfortunately, as much as I'd love to continue, we're at 8:30, so I wanted to thank very much our panelists here for really being great participants in providing us a wealth of information. I think they deserve a round of applause.